Hello and welcome to Chemspec Digital. I have the pleasure to introduce the next presentation as part of the RSC Symposium on Continuous Flow. However, before I do introduce our presenter, I'd like to point out that we are recording the presentation to make it available to view on demand. Attendees are invited to write questions into the chat function on your screen, and from there we will read them out at the end. But without further ado, I will hand over to Dr. Douglas McAndrew of Intensichem, who will be presenting developments and scale-up in the field of industrially relevant flow hydrogenation. Over to you.
Thanks very much for the introduction, and thanks again for the opportunity to talk at this digital conference. So today I'm going to talk about the work we've been doing, looking at here at Intensichem, primarily focusing on scale up in the field of industrially relevant hydrogenation. So I'm going to talk about a bit about Intensichem and our interest in flow hydrogenation and how this arose. I'm going to give an overview of technology, then how we've applied this technology to a range of applications in chemistry, and also a few examples of successful scale-up that we've done. So Intensichem was formed to explore flow chemical manufacturing in 2011. The founder of the company was Oliver Townes, and a bit on his background is that he was ex-Pfizer. There, he was the site PAT champion and also the site new process technology champion. He was also on the new technology steering team, and during his time there, he was a global technology and innovation award winner. So I knew Ollie at Pfizer. I joined him again about 3 years ago. I worked in both manufacturing within then, and also I worked in the R&D department in Pfizer. My background is mainly manufacturing support and process chemistry, and during my time at Pfizer, I won green chemistry awards in both manufacturing and R&D departments. Another key person that's worked on flow hydrogenation here is Wallace. She's also ex-Pfizer, and she spent 20 years in hydrogenation research there. So a bit about flow hydrogenation. Most people will have heard about the Thales Nano H-Cube, and this system came out in 2005. Ollie started building systems when he was at Pfizer around 2006. Again, another company that are involved with flow hydrogenation were HEL, and they made the flow cat around 2008. The hydrogenations are very common reactions. Depending on the literature reference, you read they're either 10%, 20%, 25% of all reactions, depending on the criteria for fine chemical pharmaceutical research. And that's why all of the top 20 pharma companies have these lab systems and use them for hydrogenations. Intensichem have focused on the scale of these hydrogenations, as this still remains an industrial challenge. So a bit of background as to how we got into hydrogenation. Intensichem were part of the AMSCI Rare Disease program in the UK, so this was advanced manufacturing. As part of that consortium, companies like GSK and AstraZeneca expressed an interest in flow hydrogenation development. So this led to Intensichem doing work on building some of these units, and Intensichem did build a pilot unit and subsequently took this out to a manufacturing plant in Spain, where it was successfully commissioned. And this is capable of large-scale production, around 1 ton per month. The following year, in 2017, there were requirements to double the capacity, and this was easily done by doubling the flow rate and the length of the reactor. Again, Intensicchem's interest in this area continued, where we've looked at a commercial 2-stage telescope hydrogenation in 2018, and we also started a project that was Innovate UK funded in 2018 to look at the applications and explore some of the chemistry, and that's work I'm going to talk about today. And we completed this Innovate UK funded project successfully in 2020, and this has subsequently led on to patents being approved in a number of jurisdictions in 2021. So a bit of background around the technology. So all the systems I described have this sort of generic flow setup where we've got the substrate in the reservoir as a solution. It's then pumped in along with the hydrogen gas. It's mixed and combined. There's catalyst in the reactor, so you've got substrate with hydrogen meeting catalyst in the reactor. At the end, you've got a back pressure regulator, and once the reaction's done, you're collecting the product in the solution in a collection system. So we find that we get good scale up here. We focused on the scalability of the flow hydrogenation, and our scale up is done because of the efficiency of these systems and the really good, the excellent mass transfer characteristics. Scale up for us is we use the length and cross-sectional area and effectively eliminate mass transfer at scale. So some of the equipment we use to do this, we primarily start with a lab development tool, which is the rig pictured in front of you. The capabilities of this equipment are that it can carry out reactions up to 200 bar. Again, we can go up to 250 degrees Celsius, so much higher pressures and temperatures than conventional batch chemistry can see. The flow rate of this system, again, is up to 15 mils per minute. We get automated sampling with this system, and this system we use to focus on flexibility and reaction understanding. So we'll do our proof of concept work with this equipment and also do some optimization work with DOEs to study the effects of the various parameters and optimize reactions with this. Subsequently, this leads us on to our scale-up system. So again, this system fits very nicely into a fume cupboard. It's manually operated. Again, it's got the capability to operate at these high temperatures and pressure, so we can operate up to 200 bar. Maximum flow rate for this system is 200 mils per minute, but that again will convert a lot of material. That would equate to around 288 liters per day. We can go up to 200 degrees Celsius plus with this equipment, and it's got a 250 mil reactor volume. Again, the importance here is that we precisely control the parameters and we don't have any limitations on the hydrogen supply. This ultimately led to us being able to deliver a full-scale plant, which is a project delivered 5 years ago. This project was for drug manufacture. Again, the selectivity we can obtain with these systems is important for APIs. So we gained a lot of learnings from sort of scaling up this plant and commissioning it, and that's given us confidence in scaling up of these hydrogenations. Again, we got a really good understanding of the system dynamics during this time, and again, flow hydrogenation lends itself very well to online analytical technology so that you can understand exactly what's happening in real time, automated production. Again, we have developed a system for scale up to a great level. So pictured on the right here is a picture of a GMP skid unit we're developing. This has got the capability of delivering 4 liters per minute solution flow rates up to 170 bar. It's fully ATEX rated for explosive gases. Again, it's got online wavelength UV detection. So again, the disappearance of chromophores, we get good understanding of how well it's working in the reactions, and appreciation of any changes there. Automated sampling as well, and it's PLC automated as well. And in the table below, you can see just how efficient these sort of units would be. I mean, starting from our lab development, we can do 1 mil a minute, but that will generate quite a lot of material for the initial studies. But this unit here at 4,000 mils per minute, if we operated that 10 months of the year and assuming a 30% weight volume substrate, this unit alone would be capable of producing over 500 tons per annum. Why is this system scalable? Well, part of when we designed this, it was designed with scale up in mind. So we ensure we have a stable fixed bed, and we do this with precise control of the catalyst morphology. We also ensure that our heat exchangers are separate from the reactor. So the surface area to volume, which is often an issue in scale up, is irrelevant here. We're not relying on the surface area of the reactor for the heat exchange. Again, we operate in a thermal window, so everything of that substrate and solution stream is exposed to the same conditions as it goes through the catalyst reactor. So it's a reaction that's over in seconds because of the really efficient mass transfer. When you compare this with batch processes which take hours, you've got the heat up and cool down times associated with batch, and that's when a lot of impurities can form, and so we can gain much greater selectivity in these reactions by this sort of tight operating conditions and quick reactions in and out of the reactor. Again, part of the key to our system is that we use very small particle size catalysts. This gives us a really enormous gas surface area. All of these reactions, as I mentioned, are over very quickly, seconds, as opposed to the hours that it takes in batch. And this is due to the sort of efficient, really efficient mixing. The gas, it gets exactly to where it needs to be. It's reacting with the substrate. So really good thermal communication for the gas, liquid, and solid in these systems. Again, by using these small particles, we found we can get really good tight distribution, tight residence times for the reaction, and low differential pressures across the catalyst bed. I mentioned there less than 10 bars. We're typically operating around 70 bar when we're studying these reactions. But during the course of the work I'm talking about in this Innovate UK funded project, we were seeing differential pressures of only 1 or 2 bar. It's important that we get precise control over absolutely every critical quality attribute parameter here to ensure that we get really good control and understand what's going on in these reactions. Another advantage that the flow hydrogenation systems have is that the conventional batch reactors tend to be huge, have very large vessels. You've got all this hydrogen going into these systems, so there's a high explosion risk. And again, they need to be sited in bunkers. So that these hydrogenation vessels for batch are not cheap, and that cost I've seen in previous literature has weighed on the viability of certain processes. So the advantage of flow here is that our reactors are small. There's very little hydrogen in the system at any one time, so there's minimal risk of explosion. And again, as you can see from the picture of our reactor for that 4-litre minute GMP skid unit. The reactors are much smaller, but can produce that huge tons of material. And these would be easily contained, these reactors, if it was required, with bomb-proof safety enclosures, if that was required by the client. So a bit about the chemistry that we've explored with the systems. Starting in the top right-hand corner and moving clockwise, we decided initially to look at nitro reductions, as these are popular reactions in the agrochemical pharmaceutical industry. They're often explored in the literature. But we decided to look at what selectivity we could gain with our system. So our initial work studied halo nitro reductions, so that we were converting the halo nitro aromatic to the halo aniline. So X there is a chlorine, bromine, or iodine. Moving clockwise from that in the top right-hand corner, we've also looked at some furfural reductions, the furfural alcohols using these furfural derivatives. That's not what I'm going to talk about today. Clockwise again, we looked at pyridine reductions. So the pyridine to piperidine ring reductions are also commonly looked at in the literature. The piperidine ring stones are seen cited as the third most common ring structure for drugs. So very popular reductions again, and I will talk a bit about them. Similarly, there is the reduction of aromatic rings to the cyclohexyl derivatives. Further to this, we've also looked at some aryl hydrodechorination reactions, and they go very well. Also some hydrogenations to dehydroxylate systems. Again, we've done work on that, and then moving clockwise down to the benzyl D protections. So we've done those with both oxygen and nitrogen to reveal the alcohol or amine, and I'll mention briefly about those. One of the other reactions we've studied is reductions of aryl esters, aryl alcohols, and they would be good to avoid some of the use of these metal hydride reagents. So we initially started this Innovate UK funded project with selective halo nitro reductions. So the first candidate we selected was 2-chloronitrobenzene, and we did some initial scouting around the various metals and temperatures, and ultimately we were getting some issues around the hydroxylamine formation, which is an intermediate in this reduction, and also over reduction, but we were able to quickly find conditions which gave good selective reduction to 2-chloroaniline in THF and methanol. Part of the issues were the hydroxylamine intermediates. So iron and vanadium catalysts have been used to help disproportionate that intermediate. In previous work, but we found that we could get good results with the right conditions just using undoped platinum catalysts, particularly platinum on silica. So having successfully demonstrated selective reduction to the 2-chloroaniline, we decided to look at some of the other halides, and I thought this would be more challenging with the bromo, but actually it worked pretty much straight away. So reductions of 2-bromo-nitrobenzene, we were able to cleanly reduce that to 2-bromoaniline in methanol using the same platinum and silica catalyst, again avoiding this intermediate on the right, which is the aniline, the over reduction there. And we found that we could do this not only with the ortho-bromo, but also the meta and the para were very cleanly and selectively reduced to the corresponding bromo anilines. And it's pleasing that we can do this with the platinum on silica catalyst, as I think previously some of the reductions have been done with rhodium, which is a much more expensive metal. I think rhodium's almost an all-time high price. We extended this work also to look at the iodo, but here the iodo-nitrobenzene was too labile under the conditions we explored. Fortunately, a quick look in the literature identified that 4-iodo nitrobenzene had been cleanly reduced using sponge cobalt, and we tried sponge cobalt with this and found pleasingly it worked under a range of different temperatures and flow solution flow rates to very cleanly give us the iodo aniline as well. We'd also seen in the literature that some other companies had looked at nitrobenzene and looking at 10% weight volume in methanol. We found it also that our system very cleanly gave reductions of nitrobenzene to aniline at the similar concentration. And again, we extrapolated these studies of nitro reductions to the ortho and para nitrophenols to get these 2 and 4 aminophenols. Again, that 4-aminophenol is of interest. It's an intermediate on the way to paracetamol. Generally, these anilines find extensive use in the chemical, pharmaceutical, and agrochemical industries. So that's why this was a good project to sort of start our investigations of the versatility of the technology. And again, these nitro reductions, because they're so exothermic and there's these intermediates which are reactive and unstable, anything we subsequently looked at we thought would be easier to achieve than these halo nitro reductions. So one of the other systems we did study, as I mentioned, was this pyridine ring reductions. Again, this is due to the popularity of these pyridines in the pharmaceutical industry. So we found we could very cleanly reduce pyridine to pyridine with sponge nickel. Again, a sponge nickel catalyst is good at reducing ethyl nicotinate to the ethyl nipactate. And we'd also seen that in labs at Cambridge in Steve Lay's group, they looked at the partial reduction to give this alpha beta unsaturated ester, shown pictured on the right hand side. We found that we could cleanly achieve that also with really very good selectivity, about 92% selectivity for that intermediate using a palladium or an alumina catalyst. So we also looked at some other pyridine type systems. 2-picoline, we found we could cleanly reduce with rhodium on silica catalyst. And we also applied this to lutadine and looking for a selective reduction, we went for quinoline to the 1,2,3,4 tetrahydroquinoline shown below. Again, you get reduction of the ring width of the pyridine ring system rather than the other AR ring. We found that would cleanly reduce. We also looked at benzylic deprotections. Benzyl deprotections again find extensive use in the pharmaceutical industry. It's a popular way of revealing functionality for further reactions or just to tidy up at the end of an API synthesis. We looked at some of the classic model reactions. So 4-benzyloxane phenol and then phenol-benzylamine have been studied as sort of these classic benzyl deprotections. And we found certainly with the 4-benzyloxyphenol, we've got very clean deprotection in methanol at 90 degrees Celsius. Again, 10% volume concentration for that reaction. So the reactions shown below where we generate the hydroxyquinone and the toluene is revealed as well as reaction. So we've done that for both the O and N benzyl deprotection. During this project, we used a range of metal catalysts, so wide variety of precious group metals, as well as more common other non-noble metals, whereby we've used nickel, rhodium, some ruthenium systems, etc. And we've also used some bimetallic catalysts. Usually we're using these on supports such as alumina and silica, but we also had catalysts with titania and zirconia. And again, an advantage of having this range of catalysts and the technology with our system means that we can access the higher temperatures and pressures than conventional batch processing. And this gives us these novel process windows so that we can maybe do different chemistry from conventional batch and use metals more sparingly. As part of the going towards scale up, we looked more at the 2-bromo nitrobenzene reduction. So we studied this in more detail and took samples over for 6 hours here. And you can see that we get excellent cell activity and reproducibility here with this. It was good for this system, and again, from more recent work, we found that we can get good reproducibility over days and weeks before any catalyst regeneration is required. Sometimes we don't need to do catalyst regeneration, but when we do need to do catalyst regeneration, for instance, when we're seeing a loss of performance, we have various ways of recovering the catalyst activity. And these ways have been successful in restoring the activity of rhodium catalysts and palladium catalysts more than 30 times. In our experience, which, given the high price of rhodium and also palladium, is good to know. Again, similarly, we've also regenerated nickel catalysts, and if required, our commercial systems would automate regeneration so that we maximize the catalyst lifetime, ensure we get the good selectivity required. So when we went into the scale up, we chose the 2-bromo nitrobenzene as a good candidate to demonstrate the selectivity. So starting from our 2 ml per minute that we were doing for the small scale sort of unit that I showed at the start, and then into the pilot unit, we extended that up to 120 mils per minute. So from 2 mils to 122 mils per minute to 120 mils per minute solution flow rate, maintaining the excellent selectivity. Again, we also did the selective reduction of quinoline to 1,2,3,4 tetrahydroquinoline, and again a similar large scale up from 0.5 ml per minute to 100 mils per minute solution flow rate in the first two pieces of apparatus. So pleasingly, those reactions worked very well and scaled up almost this 100-fold. So in summary, I'd like to say that we've shown that we can successfully scale up these flow hydrogenations. Here we've had excellent selectivity achieved and high yields, and I've talked about the advantages of doing these reactions quickly and minimizing the times for heating up and cooling down, and also that we can rapidly explore a range of conditions to obtain the selectivity we require, which gives us the high yields and the low levels of impurities. I think flow hydrogenation does offer significant advantages in that it's safer. We get this greater selectivity and more sustainable processes are achievable, and I think that's critical at this time. that we are developing better ways of manufacturing chemicals. And again, I've demonstrated the versatility of this system in exploring a wide range of chemistries and functionalities. We've looked at a whole range of chemistries, including reductive aminations and nitrile reductions as well. And so this other work here as well that we've been doing. And the equipment has demonstrated that it can very well achieve excellent yields for hydrogenation and definite advantage. So I'd like to acknowledge the help that we've been given in developing these systems. So this project started off as part of the Armstead Remedies project, and so there was funding available from the Department of Business, Innovation and Skills. And more recently, the work that I've talked about, we was supported by Innovate UK, and that was very welcome for all their help as well. So I'd like to thank them and you for your attention, and ask if you've got any questions.
Okay, thank you very much. Yes, we do have a couple of questions that have come in from the audience. Now, forgive me, I'm hoping I'm going to get this right. There's a couple of questions within the actual chat. For somebody who doesn't know anything about hydrogenation, does all the H2 react? And if not, where does the supplement of H2 go which didn't react? How can you manage that? I'm hoping I got that question right for whoever asked that in the audience.
So the hydrogen, usually with these systems, there is an excess of hydrogen. You're looking for the hydrogen to be bubbling out at the end of the system. For the lab scale units, these can usually it's just safely vented through the fume covered system. So we're operating these in a fume covered, and certainly our system, the flow rate in the fume covered is able to deal with the sort of excess of hydrogen that we're using. And again, that's seen in similar systems in other lab systems as well. So, yeah, we do have some ideas around handling of the hydrogen as well, but typically in the lab scale units we have hydrogen.
Okay, and the other thing, the other question was, you gave the flow hydrogenation reactors for lab scale, but what are the commercial ones for industrial scale?
So we have put one of these units out at a plant in Spain, and so I can't give you too many details on that because that's for a customer. That there was that picture of the 4 liter GMP unit that is capable of producing a huge amount of material. So that would make up to 500 tons. So we generally, a customer will come to us with a project. We'll do some proof of concept work if required, and optimization work if required, and then we'll cater for their process with the development of bespoke equipment for their process. So the reactors that we assemble, we take the cue from their processes to what materials of construction they'll require. Again, whether their system will require regeneration, etc. So it's very much designed with the customer's process in mind, so that we will develop equipment for them. For that, I hope that answers the question.
Yeah.
Please get in touch with us to talk more about the systems.
Excellent. Thank you very much, and thanks for adding in the questions. That's very, very good. I think this actually marks the end of our session. At this point, I'd like to thank our presenter and for everybody watching, and wish you all a great afternoon at Chemspec Digital. Goodbye.